Welcome back to the lecture series in animal cell culture. So, today we are into the fifth lecture of week 3. So, if you recollect, in the last lecture where I ended was a very interesting point, that every cell secretes an unique extracellular matrix. Think logically for a minute, that will make more sense to you. So, in our body, we have this heart, there are heart cardiac cells growing in that location. We have the brain, where the brain cells are growing in that location. We have muscle, skeletal muscle, where the skeletal muscles are growing in that region. And each one of these have certain different mechanical properties. Right? Say for example, if I talk about the mechanical property of the heart, they are entirely different than the skeletal muscle, than the mechanical property of these cells in the brain. I am just taking the simple ones, and you could have many more. You can think of the cells which are lining the gut, where the food is moving through the smooth muscle. What do you mean by the mechanical property? Okay. Your heart is beating, so it means cells are moving like this, right? Now, having said this, think for a minute. Just this is something you really have to say. For example, these are your heart cells, okay? Okay. Now, just for simplicity's sake, I am just putting it like that, and these cells are suspended. The blue is some kind of extracellular fluid which is there, and these cells are anchored to each other by extracellular matrix, as well as to the surrounding tissue by certain degree of extracellular matrix. Okay. Now, while this cell is moving, so there are different kind of movement which this cell is undergoing, right? So, it has movement like this, it has contraction like this, it has contraction like this. So, every time while it is doing so, there are movement in these attachment points. You see, these attachment points, it is like these attachment springs or attachment points are getting stretched and strained all the time. So, coming back to the drawing, what I was trying to tell you, see for example, this stuff is kind of have a flexible arm to move around. Now, imagine for a minute the force generated by this attachment point is higher than the force with which it is attached. Because of this contraction motion out here, the force generated by this attachment peg is higher than the adhering force. So, there are two forces here. One is the force generated due to the movement of this part. Let us draw it simple, much more. Okay. Okay. This is how it is attached, okay, and this is the cell end. So, this is the point of attachment out here on a substrate, and this is the substrate. Okay. So, there is one force which is helping it to attach, attachment force. Okay. There is an attachment force, whereas there is another force which is functioning here, which is force generated due to contraction mechanical activity of cell. Now, if I denote this force with F prime and I denote the attachment force as F double prime, and if F prime in terms of the Newton is greater than F double prime, then what will happen? This cell will get detached from this surface. So, in order for this cell to remain there, either it has to be intacted in such a way that it kind of adhere there, or its mobility has to be flexed in such a way that you give it a linear window to flex like this. So, it means this attachment point has to be equally a flexible surface. You see my point? So, instead of, so here what I am telling you that you have a glass substrate, right, which does not have any additional molecules or anything. Now, on top of the glass substrate, you decorate this glass surface with something like this, and once your cells come and sit there, cells are coming and sitting there. Now, these cells are secreting something which kind of bind like this. So, this, let me change the color, that may confuse you. Let me use it as red, okay? That will make more life more easier. So, this is the red which you have coated the surface, okay? And the pink one, what you observe is secreted by the cell. So, now if these two interact with each other, now, so it is something like this. This is the molecule I have coated the surface, so it is dangling like this. Here is a cell coming and it is secreting this arm, which is, now I am waving this arm. This is the arm which has been secreted by the cells. So, these two come and form a bond like this. Now, you see they can move better like this. So, this is precisely what I am trying to tell you is, whenever we talk about the substrate vessel and modifications and all this is surface modification, this is precisely what can be done. But then, having said this, I will ask a different question before I get into this. So, I told you, so different cells at different places. So, I took example of 4 cells, okay. One I told you about cardiomyocytes, one I told you about skeletal muscle, the third I told you about smooth muscle, and the fourth was about neurons in the brain, right, because the smooth we added. Now, all these different muscle type and the cell type, the neuron, since this is neuron also, so I cannot call all our muscle type. Even within this muscle type has different mechanobiology. Their mechanical interaction with the surface is different, with its surrounding cells is different, and it generates different kind of forces out there, which is governed by, this force generation is governed by the cellular chemistry of extracellular matrix materials. So, there are two things. One, its mechanical properties varies. Second, the mechanical property is the function of cellular chemistry of the ECM material. So, it seems, if we look at it very carefully, it seems these cells prefer to remain in different kind of colonies of their own with a different kind of surface interaction. So, if you have to culture a specific cell type in a dish, you have to have an idea about the extracellular matrix, what or which will help that cell type to mimic or to feel much of the same environment where it is growing in vivo or in real life. This is one aspect. So, if you know that extracellular matrix nature, then what you can do here, you take the substrate. So, here you have the substrate. So, you wanted to say grow cell 1, cell whatever, then you coat that substrate with that choice of your extracellular matrix protein or material, because it has carbohydrates too, like this. Now, modified situation, here is your cell coming. First of all, started to roll, it has its medium out there a little bit. First, it looks for its electrostatic interaction, positive, negative interaction, which it will do, it will fall in a substrate, and then this possibly, this is something which is believed to be, this extracellular coating what you have done may send it a signal through its surface receptors to its DNA, telling it, dude, I need some of those proteins to be secreted by you, which are going to form bond or will start dating with this other protein which is on the substrate. Now, you see, so now they are kind of cross talking with each other, and this is how a cell initiates its life outside the system. What you are essentially doing, you are taking the cell from its home, you are marrying the cell, getting it married to another environment, another substrate, another house, and your house is that vessel where you are getting them married. But now, if you want them to succeed there, you have to create a conducive environment for them to feel at home. So, this conducive environment for them to feel at home is out here. Yet there are some cells who, why they do, we do not know, they prefer to lose their extracellular harmony and can grow anywhere, any community, and can start to proliferate anywhere. They are kind of rogue cells. You have seen this movie called Rogue Nation, something like a rogue cells. These cells absolutely can go anywhere. It means they can either adapt to the extracellular environment of anything and everything, or they do not even need one specific one. And these rogue cells are or is the beginning of what we call this word, cancer. So, those cells this is another way you can define cancerous cells. Those cells whose extracellular matrix harmony is compromised, they travel along wherever they can and develop the rogue colonies there. And what they do, from that surrounding, they pull out the resources for their own benefit and pretty much cripple that community and make them devoid of their necessary requirements and eventually ensure that they succeed till mankind succeed to understand their own problem inside them, that they have such rogue nations or rogue cells within them. They can do wonders. Now, that brings us to a very different paradigm altogether. The paradigm is this. This has been there for a long time and people are contemplating over it. The paradigm is this, that if we accept for a minute that this is how it is working, so it means when the sperm and the ova fertilized, when a man and a woman had sex and they have a baby conceived. So, the first cell which is formed was a zygote, right. So, you have this sperm, one second, let me take this. So, okay, this is sperm and this egg fertilized with each other and what we call as a 2N or a zygote, right. This all of you at up to class 10 somewhere or other you must have studied. This zygote then form a mass of cell and this mass of cell eventually form an embryo and this embryo eventually becomes a baby. Now, think of these phases while they are going through. These cells, there is no colony because it's still with very early phase. So, these cells were adapting to different extracellular matrix. At the initially they were all clustered together. It means the cells from where we are originating, they clustered together with that unique extracellular matrix and then eventually the extracellular matrix differentially, like cell community of cells, like 10 cells will secrete different kind of extracellular matrix, another 10 is going to secrete different extracellular matrix. But earlier to that, if you think of this situation where they could attain any kind of extracellular matrix fate, aren't they similar? Aren't these cells similar to these cells? So, that brings us to a very different way of looking at cancer. Is it a developmental disorder or maybe not by the right word, because developmental disorder is used for a very different connotations, but it is something the answer may be lying in developmental paradigm. Let us put it like this. Instead of borrowing any word from anybody, let us put it like this. Because those cells, try to just visualize a single cell become a mass of cell. These mass of cells eventually pick up their colonies. Some become red, some become green. I mean, just here I am putting it symbolically. Ooh, some becomes violet, okay, some becomes yellow likewise, okay. So, one second, okay. So, these colonies while they are formed, earlier to that it was they can do anything and everything. Are they similar? The questions which future is going to answer, but at this stage what is very, very important for you is to understand this. These are some of the very emerging areas which are coming up. This mechanobiology, and a great understanding is happening in the cellular chemistry of ECM materials. So, the question where now we are landing up with what are those extracellular ECM, natural ECM? There is a reason why I use this word natural ECM. Natural ECM which can be used to coat the vessels to grow the cells of your choice, one, and what are those synthetic ECMs? What we mean by synthetic ECM is, if these are your natural ECMs, you can always develop analog or something which mimics these natural ECM. Okay. So, those which mimics these natural ECM, those I termed or coined them as synthetic ECM, and we will talk about this synthetic ECM from very unique bioorganic kind of things to very inorganic kind of things. We will talk about it. So, at this stage, what is important, the take home message for you people is, yes, we have these different kind of vessels where I started with. So, we are the fifth lecture, just let me put it like, week 3, and this is our lecture 5, where we are almost at the phase of concluding it. So, we have these different kind of vessels, and then on top of that we can do a lot of modifications on them depending on what cell type we wanted to grow, and we can either go by synthetic route or we can go by natural route, and what kind of interaction they will do. And having said this about the cell type, in a very subtle manner, I told you within the adhering cell type, what cell type are we talking about? Means, is it cardiac cell? Is it cardiac muscle, or is it skeletal muscle, or is it smooth muscle? Is it neuron, and what neuron within them? Different neurons have different requirements, will come in the specialized section. Or is it liver cells? Is it pancreatic cells? So, this list can go on and on. What is important for you to understand is, individual cell types have an individual ECM, barring aside those which becomes rogue. You can call them rogue, or you can call them some reprogram themselves back to the origin. Reprogramming back to the origin. So, that is why we came up with this thing. Does the answer for cancer lies in the developmental paradigm? But that also tells us something, that could we create a matrix to trap the cancer cells? Say for example, we know here you have the cancer cells and they have a competitive binding for a substrate, and as soon as they hit on that substrate, you can program the substrate like that, it will induce death in them. Could we have such things? Could we think a little louder, much ahead of our time? That could make a whole lot of difference. I am looking at cancer from a surface chemist point of view. So, with this thinking, I will close in this lecture. Next week, we will continue this story. Thanks a lot.